Greeting friends, this is the third video in the lecture series of membrane proteins, and here I'm going to talk about the third category of membrane proteins, which are known as peripheral proteins. Now why they are known as peripheral proteins? Because they are present on the periphery of the plasma membrane, and we also call them as surface proteins because they are present on the outer and inner surface of the plasma membrane, that is the outer leaflet and the inner leaflet. We also call them as extrinsic or external proteins because they are present on the external surfaces of plasma membrane, like we named the transmembrane proteins as intrinsic proteins or the internal proteins because they were spanning the membrane through their hydrophobic segment. Now there is an important difference between these peripheral proteins and other two categories of proteins, that is the transmembrane proteins and the lipid anchored proteins. So just focus on these two diagrams here, the first and the second case. Let me draw something here. So you can see here this is the spanning segment of the transmembrane proteins. Through their embedded segments, they make a direct contact with the phospholipid bilayer, which is composed of the fatty acid chains. So these are the fatty acid chains. And if we talk about the lipid anchored protein, I have not shown here. Let me draw the structure first. So basically the lipid anchored proteins are, the lipid is embedded in the one of the monolayer, okay, and this lipid is covalently attached to a protein moiety. Say this is present on here. So this is a protein moiety, okay. So we can say here, in case of these lipid anchored proteins, with the help of this attached lipid, it is making a contact with the plasma membrane hydrophobic core, and through their spanning segment, these transmembrane proteins, they make direct contact with the phospholipid bilayer. But this is not going to happen in case of these surface proteins. So if they are not making any contact with the phospholipid bilayer, so how they are going to interact with the plasma membrane? That is the question now. But before we come to that, how they interact, I forgot to tell you something here about lipid anchored proteins. This one, we know there are three categories of proteins. There is transmembrane proteins, and the lipid anchored proteins are the surface proteins, which we are going to discuss here. The thing here is that these lipid anchored proteins, sometimes we also call them as surface proteins, because if we see clearly, this one, the protein, this is present on the surface of the plasma membrane. So we also name them as surface proteins. Why? Because they are present on the surface of the plasma membrane. Now why they are categorized under lipid anchored proteins? So this is a different category because they are anchored with the help of a lipid. So my point here is to tell you this thing is that, because sometimes in your exams, examiner may ask you the question there, which of the following regarding lipid anchored proteins is correct, or directly it is given that sometimes there is a question. I have noticed that there was a question regarding GPI anchor, in which it was asked that which of the following statements are true regarding GPI anchor, and GPI anchor is present in the extracellular leaflet of the plasma membrane, and that anchors the basically the protein to the extracellular leaflet. So there were nothing about the anchoring of the proteins with the help of that GPI anchor, and the option correct was that it is a surface membrane protein. So you have to remember that they are also the surface proteins, the peripheral proteins. So that is the point here, okay. So now let's know that how they interact with the plasma membrane if they are not connecting to the phospholipid bilayer. So for that, there are two ways, okay. Through two ways, these proteins, the surface proteins, they can interact with the membrane, that is the indirect way and the direct way. Before we go into these ways, let's assume something, okay. Let's assume something. There is an assumption that there are two guys. One is boy with this blue color, say. So this is a blue color boy, and there is a green color girl, okay. Now this girl likes this boy. She has feelings for him, but the problem here is that this girl is too shy to talk with that boy, or she is hesitating to interact with this boy. But so for this thing, so for the friendship, what she does, she asks one of her friends, say this red color friend is coming here now. So she asks this boy, can you please help me to talk with that boy? I really like him. So now this friend is going to help help her actually. So now there is going to be a friendship. So we can say this lol. Okay, so now there is a friendship. Now assume another case. Now suppose the girl is not shy, she is too confident. She says that I can talk to that boy directly. So this is the boy, and there comes the girl who is confident. So she says that I can talk to that boy directly. So friendship is occurring. Okay, that is it. Okay, so let's apply this assumption to these proteins. We can say this is the protein which is the surface protein in green color. So due to some problems this cannot interact with the plasma membrane directly. So what this protein is going to do is that this is asking this transmembrane protein, because this is present in the cytoplasmic layers, so we are talking here about the cytoplasm. Okay, so this again occur in the exoplasm as well. So this is asking this protein domain in basically the cytoplasmic domain, that I cannot interact with the membrane, can you please help me? So this protein here is helping this surface protein to interact with the plasma membrane. So this is the indirect way where this domain is helping the protein to interact with the plasma membrane. Now the direct way where this help is not needed, in this case the protein is capable of interacting with the plasma membrane directly. So how this protein is going to do that? So that's the question now. So we know the phospholipids are made up of two things, basically the fatty acid chains this way, and another thing here is going to be the phospholipid. So this protein interacts directly with this phospholipid. So that is the direct way. So we have finished the indirect and the direct way of interacting with the plasma membrane by the surface proteins. So after this we are going to discuss some functions of these proteins, the surface proteins, important functions we can say. First, at outer leaflet, so they are present on the either side of the membrane, outer leaflet as well as the inner leaflet. So at outer leaflet what's going to happen? So many times you are going to see that the proteins which are present on the extracellular leaflet or the outer leaflet, the surface proteins, they are going to attach to the extracellular components of the extracellular matrix. So the components which are present in the exoplasm or the extracellular matrix, these proteins, they attach to them. So if this thing happen, what is going to result? So these proteins, this way they act as a crucial interface. So this is about the interface. They act as interface between the cell and its environment, and if this happens, this results into the involvement of the signal transduction pathways. So what does this all mean? Let's draw through an example. Let's see this through an example. So what does it say? That if this is the cell, assume this is to be a cell, okay, and there is something like in pink color, this is not specific, just assume that this is something in the extracellular exoplasm that is in pink color, okay. Now there is a protein which is the surface protein which is present on the surface of the cell. This is that protein, okay. So this protein, this surface protein, is acting here as interface between the cell and the attached extracellular component, and through this attachment, this extracellular component and this protein, they are executing some important signal transduction pathways. You would study them in this signal transduction topics. And now we talk about the functions at inner leaflet. This is going to be same. The difference here is that this is occurring in the inside of the plasma membrane, that is the inner leaflet. So we know about the cytoskeletal proteins. What are those? Like actin and spectrin. There are many more. we are not interested about them here. Actually, the cytoskeleton proteins, these are the one which make the frame of the cytoplasm. Now these proteins, they have a connection with the enzyme PKC, protein kinase C. So this PKC actually is a binding partner for surface proteins. So what does that mean? So let's see. This enzyme actually, it is present in cytoplasm, but in inactive form first, and when a signal is coming in signal transduction pathways, this shuttles from the cytoplasmic side to the cytoplasmic leaflet of the plasma membrane, and here it attaches to a one of the surface protein, and this mechanism is going to execute some important functions you would study in signal transduction pathways. So this was regarding the membrane proteins in surface proteins. And in the next videos, I would talk about the specific examples of the proteins and the structures of the transmembrane alpha helical conformations. Okay, thank you.